We know that DNA is a double-stranded molecule. Its two strands are held together mainly by hydrogen bonds. Inside cells, enzymes called helicases separate these strands during DNA replication. But in the laboratory, DNA strands can be separated either by heating the solution or by changing its pH. When DNA is heated, the hydrogen bonds between complementary bases break, causing the double helix to separate into two single strands. This loss of DNA's helical structure is called DNA denaturation or DNA melting. Remember, only the hydrogen bonds break. The strong covalent bonds of the sugar phosphate backbone remain intact. As the temperature continues to rise, more and more DNA molecules become denatured. However, this transition does not occur evenly over all temperatures. Instead, most DNA molecules melt rapidly within a narrow temperature range. The midpoint of this transition is called the DNA melting temperature. It is the temperature at which half of the DNA molecules are double-stranded, while the other half have separated into single strands. But how do scientists determine this temperature? DNA bases naturally absorb ultraviolet light, especially at a wavelength of 260 nanometers. In double-stranded DNA, interactions between stacked bases reduce this absorbance. As DNA denatures and the bases become exposed, they absorb more ultraviolet light. This increase in absorbance is known as the hyperchromic effect. Using a spectrophotometer, scientists measure the absorbance of a DNA solution at 260 nanometers while gradually increasing its temperature. When absorbance is plotted against temperature, a sigmoid-shaped melting curve is obtained. The lower part of the curve represents mostly double-stranded DNA, the upper part represents mostly single-stranded DNA, and the midpoint of the curve corresponds to the melting temperature. If the DNA solution is then cooled slowly below its melting temperature, complementary strands recognize each other, reform hydrogen bonds, and restore the double helix. This process is called renaturation, reassociation, or annealing.